Okay, let's start. So I have the pleasure to introduce today our dear guest Frank Postberg. So it's one of our early department seminars, and before he really gives very likely a very exciting lecture, I will briefly introduce him and of course welcome him here in my new home here in Prague at the Heyrovský Institute. So Frank did study chemical engineering, and he did a diploma in physics back in 1998 and 2004, but he did his PhD in the Faculty of Physics and Astronomy at Heidelberg University. And at that time, and later on, he was research group leader, and I think we met at 2007 or something like that.
2008, I think.
Still did your PhD thesis, but that was a sort of exciting time when not the first dust grains, but dust grains were analyzed. And then after diploma and PhD, he did his habilitation at the Faculty of Chemistry and Geoscience in Heidelberg University. He was visiting scientist with the German Science Foundation Heisenberg grant in the Laboratory for Atmospheric and Space Physics in Boulder, Colorado. And since 2018, he's professor in planetary science in Berlin, Free University of Berlin. So, I think I don't want to take too much time of your time here, and I'm happy that it worked out, and the stage is yours.
Thank you, Bernd, and thank you, Jan, for inviting me here. I'm excited to be the first time here in my life, but I'm sure I will be a frequent visitor in the coming years. Because we have already many collaborative proposals specifically in the last 5 or 6 years. And now I give a very general overview, because I know most of you are not familiar with planetary science, but there has been major, a new direction of planetary science by introducing mass spectrometry into the field. And the object you see here, this is Enceladus. This is like the poster child where we first learned how powerful analytical chemistry in the form of mass spectrometry is to explain what is happening in the interior of this icy moon, which turned out to have a subsurface ocean. And so I will first summarize the results we got with the Cassini spacecraft and the mass spectrometers on board for Enceladus. This mission is already over. We are still analyzing data, but the mission is over. And now we are in a stage to prepare for future missions. And this will be the second half of the talk. So first of all, to acknowledge the significance of these icy moons is that in 1997, we found that Mars is dry. There was hope that there was liquid water on Mars, or even in the 60s on Venus. No, very dry, no liquid water. And then people gave up actually hope to look for life in our solar system. Like 30 years ago, when the data from Venus and Mars was there was very little hope because there was no liquid water. And this changed dramatically. Now we have in our solar system alone 6 to 11, depending how strict your criteria are. There are at least beyond Earth at least 5 more generally accepted bodies with liquid water in the solar system. And these are they all look like this. This is Europa, Jupiter's moon Europa, an icy moon. That was the first one where the subsurface ocean was discovered in 1998. And since then we have at least 5 more. So 3 orbiting Jupiter, Europa, Callisto and Ganymede, and 2 orbiting Saturn, Enceladus and Titan. So you see already Enceladus is much smaller. Europa is about the size of our Earth moon, and so Titan, Ganymede, Callisto are larger, and Enceladus is much smaller. And so Europa subsurface ocean has at least 2 times more liquid water than we have on Earth. So most of the liquid water in our solar system is not on Earth. It's by far resides in these subsurface oceans in the outer solar system. And this is probably generally the case in the universe. So most of the liquid water is not on the surface of terrestrial planets. It's probably in icy moons orbiting gas giant exoplanets as well. And there's actually a new kid on the block. So minus about a year ago, the data is borderline that conclusive. Most people agree that Mimas, it's a small moon like Enceladus orbiting Saturn, also has a subsurface ocean. And there are 3 or 4 more candidates like Pluto, Triton, the moon of Neptune. These 2 are already very hot candidates to have subsurface oceans. So that's a total paradigm change in the last 25 years. Liquid water is nothing rare in our solar system or even in the universe. It's abundant, and it's mostly in these kind of icy moons. Okay, and yeah, the poster child is at Saturn, Enceladus. Here's Enceladus. Let's go back. This is Saturn, and here's Enceladus. Here's another moon, Tethys. And this is an image you cannot make from Earth, obviously, because the sun is behind Saturn. So the spacecraft, Cassini spacecraft, took this collection of images when flying behind Saturn. And in the spectral illumination, you not only see the main rings here, but you also see diffuse dusty rings that you don't see from Earth, because they have the dust particles that make up these rings. Here it's the E ring. Oh, why does it move automatically? Are so small that they hardly reflect light, but they scatter light very efficiently. And also the color of the ring, the blue ring, tells you that there are very small micron-sized particles dominating these outer E ring. And Enceladus' orbit is perfectly coinciding with this outermost ring. And this was already known before the Cassini spacecraft. But how the E-ring and Enceladus are connected, that was then only discovered in 2005, 2006 by Cassini, namely that Enceladus is a cryovolcanic active body that actively spews out ice particles into space, and since the moon is so small, the gravity is so low that the ice particles escape, and then when the moon moves around Saturn, it constantly loses ice grains that in the end make up the E-ring. So the E-ring is basically a storage ring for ice particles that come from the interior of this icy moon. This is another close-up image where you see that there are like 4 fractures where these icy jets penetrate the icy crust from the interior. So some basic physical parameters. The moon is really small, so 500 kilometers, but the density is relatively high. So it's 1.6 times the density of water, and for an icy moon that's relatively high. Usually icy moons have a somewhat lower density that's closer to the density of water or water ice. That already tells you that this moon is not just an icy moon. There must be a substantial fraction of the moon must be rocky in order to get such a high density. The temperatures usually on a sunny summer afternoon at the equator, you get 75 Kelvin, because we are here 10 times further away from the sun than the Earth, and then what you get from solar radiation alone is 75 Kelvin. And it was even more so surprising that on this active parts at the South Pole, where these jets penetrate the ice crust, we get temperatures up to 200 Kelvin. So it's a bit, it's really a lot of heat that is emitted here, And you can calculate the power that is needed, would be needed to create this amount of heat. It would be 5 to 60 gigawatt alone. And it was for over 10 years, it was totally unclear why and how this small moon creates this energy. Not talking about the subsurface ocean. At that time, the subsurface ocean was just speculation, but just this emission of so much heat was puzzling. But the Cassini spacecraft then changed the entire mission design and sampled this cryovolcanic plumes to learn more about what's happening at the interior of the moon and why so much heat is there, and if there is a subsurface ocean. And this was then the hour of the two mass spectrometers on board of Cassini. One, the ion and neutral mass spectrometer, and then the cosmic dust analyzer that was analyzing the composition of the ice grains, and this, of course, the composition of the neutral gas that was emitted. Neither of the instrument was built for this job, obviously, but this was a very diverse, old-school spacecraft that had explorative payload that could immediately take on the new task. And I summarize here what these two instruments told us until about 2022. Later I will focus on some later results. So by the composition of the ice grains, and that was actually the first time we collaborated already together with Bernd, at that time in Göttingen, was that the model of these salt grains are salty, and from these salts we concluded that the ocean has sodium and potassium salts, sodium chlorides, potassium chlorides like our Earth ocean, but a much higher abundance of sodium carbonate and bicarbonate. That's why it's also a much higher pH because of this high carbonate abundance. Many people call it a soda ocean. Also because of the alkaline pH, and also because you have a lot of CO2 bubbling from below. That makes it fizzy. Then we find also a lot of organics in the ice grains, low mass organic species, amines and cardinal compounds. With the low resolution of the mass spectrometer of the cosmic dust analyzer, we could not specify it further, but we could positively identify these functional groups. And then we had a very different kind of ice grain where we had macromolecules included with atomic masses in excess of 200 or 400,000 even, that also we could also identify oxygen and nitrogen bearing functional groups. And then the plume volatiles analyzed by the neutral gas spectrometer. Not surprisingly, most of it is H2O, but a large, almost 1% of the gas is hydrogen, and that was really surprising at that time, because you cannot store hydrogen on such a body with such a low gravity. The hydrogen will go into space after a few days, even if you store it in the core. It is so volatile that it will escape. I mean, hydrogen escapes from the Earth all the time. The atmosphere cannot store it, so neither can Enceladus. So you have to produce actively hydrogen. That was an important finding. Then ammonia, CO2, I was mentioning already, some methane, and C2 and C3 organics. Everything with more carbon atoms is not volatile anymore. You don't find it in the gas, you find it in the ice grains at these temperatures. And tentatively detection of sulfur hydrogen. So now, before I continue, let me first introduce to you how we think now that the plume works, and how the liquid water ocean communicates via the plume into space. So the ice crust at the south pole here, I mean the moon is now upside down, this is the south pole, is very thin, maybe 3 to 5 kilometers are the latest estimates. And the icy crust basically swims almost isostatically on top of the ocean. And with that, of course, the water rises through these crustal cracks. Up to like 90% of the diameter of the crust, the water column will rise in these fractures, and with that, the water is maybe just a few hundred meters away from open space. And this is a very unusual situation that you have liquid water exposed almost to open space. This is obviously not a stable situation, right? Because you have zero pressure, cold temperatures, but you have some back pressure in the vent here that allows to stabilize it. So you have almost the triple point conditions here, maybe 6.5 millibar. That is just enough to avoid immediate freezing. And with that, but still the water is close to its boiling point. It's close to 0 degrees, it's in contact with cold ice. So it must be close to its freezing point, but it's also close to its boiling point because the pressure is too low. And with that, it evaporates a lot of water vapor that then follows the pressure gradient and is basically sucked out into space and reaches this tremendous speeds. So the gas speeds here reach kilometer per second. That's like faster than any, if you shoot a rifle, you are like at 500 meters per second. Here we're like kilometer per second gas speed. And it basically carries the ice grains with it that are ejected at somewhat lower speeds. That's how the plume works. So, and of course you have vapor condensation here. Where in these icy vents, you have some nozzle effects where you will condense some of the vapor. But there are these salty grains that carry the ocean composition. They are probably aerosols that are created by bursting of gas bubbles. We have hydrogen, we have CO2, we have methane, we have a lot of volatile gases bubbling up from below, and they will inevitably create an aerosol that then can freeze and is likewise ejected. So we see salt-poor grains that are vapor condensates, and we see very salt-rich grains which come from the liquid ocean. And this is a high-speed camera sequence from a bubble that bursts. It's totally not related to Enceladus. It's just showing you how we create these little droplets from a bursting bubble very nicely. And with the pressure gradient into space, you can then carry these aerosols into space. Yeah. Then for the high mass organic molecules, for the low mass organic molecules, we find in the ice grains they are probably just vapor condensates. But for the high mass ones, with atomic masses in excess of 200, they must be already solid at the start. And we find them in salt-poor ice grains. So the only way we could explain high mass organics in large quantities in the salt-poor ice grains is that we invoked an organic enriched film on top of the water table here. That when you and we have that also on Earth, we have a biofilm. Here we probably don't have a biofilm, but we know that bubble bursting on Earth oceans create a spray of both salty aerosols but also the organics. And probably the same thing happens here, and then the organics, they get an ice coating, and the ice coating is then, because it's from the vapor, it's also salt-free. That's our working hypothesis, how we get some of these high mass organics into space. And about 1 to 3% of the ice grains contain this high concentration of high mass organics. Okay, we also learned something about the ocean floor. Also from the Cassini spacecraft, during the flybys, measured the higher moments of the gravity field, and with that could infer a density layering structure of the moon. And they could see clearly in the gravity data the rocky core. So we know very well what the size of the core is. And the core was much larger than expected from the previous models. So in turn, that means that the density is lower than expected. It's only 2.3 gram per cubic centimeter. That was just recently updated in the 2024 paper. Before it was 2.4, now it's only 2.3. And any rock, most rocks have like 3 grams per cubic centimeter. 2.3, there are some exotic rocks that are that low density, but an entire rocky core with such low density makes only sense if it's porous, if it's not compacted. And that wouldn't work on larger moons with high gravity. So even if the moon would have like the size of Earth moon, the gravity would already be sufficient that you compact the core. The gravity would self-compact the core. But with the low gravity of Enceladus, it's only 1% of Earth gravity, that would allow porosities in the core to survive. And this was an important finding to explain the energy source of the moon, which I come back to later. So yeah, ocean water can probably percolate freely into the core. It's not a sealed ocean floor, it's a porous ocean floor, and so water can sink into the core until probably the center of the moon. And in this way, you can create hydrothermal fluids by tidal heating. I have a show how that works in the next slide. And then you have hydrothermal regions here. And so the first indications for this high temperatures at the bottom of the ocean came from the hydrogen I mentioned. It's an easy way to create hydrogen by serpentinization reactions at hydrothermal sites. And the other evidence was this silica here. That's the only uncharged species we find. Nanosilica were also interpreted as an indicator for hydrothermal fluids, because only hot water can dissolve large quantities of this quartz, basically, from the rock in a sufficient quantity that explains the measurement we did. And so we had these two measurements from the mass spectrometers, and then we had the model that also gave us high temperatures here from the porous core. Yeah, that's what I just said. The H2 emission. And there's only 2 ways you can create H2. At least two ways that are currently published. One is the serpentinization reaction, and the other is decomposition of complex organics. But then you need even higher temperatures than just by the serpentinization and mild hydrothermal reactions here. And the reason, the energy source is tidal heating. And tidal heating is something that is, it's not uncommon in space. It's uncommon on Earth. I mean, we have the tidal input of the moon on Earth is just a little bit of tides on the seas, so you have some movement in the seas. But if you have a small moon orbiting a large gas giant with a lot of gravity in an eccentric orbit, then you can basically deform the moon. And with that, you can have this deformation basically can dissipate heat. Right? Any deformation dissipates heat. And it's specifically efficient in this porous sponge-like core. The modeling that tried to dissipate this tidal energy in a solid rock only yielded 1 gigawatt. But we observe already 6 gigawatt at the South Pole, and to maintain a global ocean we need at least 20 or 25 gigawatt. But when they tried to model it with this porous core, then of course you create friction between the particulates, and then the tidal forces can be easily turned into heat. And then you flow the water through it, and then you heat up the water as well. And this models automatically with the porous core then gave temperatures here at the core of 200 degrees, 100 degrees then here at when the hydrothermal fluids, they sink into the core here at mid latitudes and low latitudes are heated up. You reach maximum temperatures in the core, and then the hydrothermal fluids are diverted to the poles where they enter the ocean as roughly 100 degrees. And you see here by the dimensions, we are at the depth of 60 kilometers here, but only at 80 bars at the bottom of the ocean. It's because of the low gravity. And even at the center of the moon, we only reached 350 bars. And so this all fit together. So we couldn't explain the hydrogen, the nanosilica, and now with this tidal modeling, everything fits together, and most people agree that this is the motor of Enceladus, the energy source. And with that, of course, you have the reason why we find so much organic material in the blue. It's probably transported upwards from the core. The core in the outer solar system is much more organic rich. Any material in the outer solar system, in any planetary system, if you form a planetary system beyond the ice line, that's where water becomes ice, and that's on Earth. It's between Mars and Jupiter. Outside that line, you always have enrichment of organics. We see that from carbonaceous chondritic meteorites, and we see that with observations of protoplanetary disk and in other stars. You always have much more carbon further away from the star when you form a planet. And this is true for organic carbon and inorganic carbon. So that's why if you form a moon there at Saturn distance, you have a lot of organics already in the core. And you can then mobilize it with this convection and then train it into a thermal plume that brings up the material here. And we think that this complex organic that we see are just the tip of the organic iceberg that we can identify with this our very simple mass spectrometer we had on the Cassini spacecraft. But in fact, if you imagine you have here water cycling through the core on timescales of like 20 to 50 million years, you have huge pressure gradients, you have temperature gradients, you have pH gradients, and you have a large rock surface, a sponge-like rock surface with a rock-to-water ratio of 5:1. Then you can imagine that this has to be an organic factory or a big organic reaction chamber where you have the conditions, different temperature, pressure, catalytic pH conditions, and gradients. Just from the flow, you introduce the gradient. And this, of course, makes this moon the most interesting object in the solar system for astrobiology and to look for life outside Earth. One thing that was missing until recently was phosphorus. So their astrobiologists find that for any organic life that's based on organic chemistry and water, you would need the six bioessential elements, the so-called SNOPs. And phosphorus until 2023 was not detected. And there was a theoretical study that got quite some attention in 2018 that modeled the chemistry of ocean moons and said, yes, ocean moons have conditions where the phosphorus keeps locked into the rock and is not mobilized. So this could be a bottleneck for life. Even though you have an energy source, you have water, you have organic chemistry, if you don't have phosphorus in a bioessential way, that means dissolved somehow, then you could not evolve life. And that's why it was such a nice starting point that then with the reinvestigation of E-ring ice grains, we found a few grains that had high concentrations of phosphates. This is now a mass spectrum of the cosmic dust analyzer. You see how terrible the mass resolution is compared to Earth standards, but clear the three phosphate peaks are a unique fingerprint. With sodium phosphate, it's at high pH because you have this hydroxide peak here. And we reproduced it, the spectrum in the lab very nicely, just with a mixture of sodium hydrogen phosphate and sodium phosphate. And we could infer a concentration for the ocean of about 1 to 20 millimole. And this is quite a lot. For comparison, the Earth has just 1 micromole. So you find this concentrations is it's like 1,000 times more, 100 to 1,000 times more than when we have on Earth. And with that, it's by no means a bottleneck for the potential emergence of life. And now we have to explain why Enceladus has so high concentration of sodium phosphates in the ocean, whereas the other models said no, no, no. But we have a solution for that. We carried out 2 years of geochemical experiments and for putative Enceladus rock compositions, and found always that the buffer system like this, maybe not exactly like this, but always similar like this, controls the phosphate concentration in the liquid, in the ocean basically here. So the reason why phosphorus usually has the lowest bioavailability is A, because it's cosmochemically the least abundant of the schnapps. All the other schnapps are much more abundant in the universe. Phosphorus is the rarest. It's not super rare, but it's relatively rare. And second, it doesn't like to dissolve in water. It's usually locked up in rock, and then therefore you say it's not bioavailable. And it's so poorly soluble because of this. It usually makes this calcium or magnesium rich minerals which are poorly soluble. But if you introduce a lot of carbonate or whatever carbonate into the system, then you can drive the reaction to the right. And Enceladus is a carbonate rich ocean. We have seen that before by first of all, we know that the outer solar system is carbonate rich, but we've found the sodium carbonates. We see a lot of CO2 bubbling out of the moon, so no doubt that there's a lot of CO2, and then you drive the reaction to the right. If you have a lot of carbonate, then the calcium is locked up by the carbonate, and then the phosphate can go into the solution. This is basically showing the concept. High carbonate lock up all the high carbonate locks up all the calcium and magnesium in the rock, and then you can dissolve a lot of sodium charge balance, phosphate charge balanced by sodium, and sodium phosphate are the only good soluble phosphate species there is. And we also find this correlation. This is this reaction, and you see how the total carbonate concentrations correlates with the total phosphate concentration in the liquid. The more CO2 you put into the system, the more phosphate you get into the solution. And this is almost independent from pH. We looked at the pH range relevant for Enceladus. And then you look at the blue region, that's the concentration of carbonates that were previously inferred for the Enceladus ocean. And then this phosphate concentration is what we got out of the data from Cassini. And this nicely match here really gave a lot of credibility to this idea. Why Enceladus not just have the schnapps, but also with an extremely high phosphate concentration. This is another way to plot the phosphate concentration, independence of pH. Here now for 2 cases, high CO2 and low CO2 case. The low CO2 case always has lower phosphate concentration. And for Enceladus, we are like at the sweet spot. Between 9 and 11 for any kind of CO2 level, the phosphate concentration peaks. So that's why we have this maximum here between like 1 and 20 millimole. And we can now, and this should be true for all the other moons that formed so far outward in the solar system. So not just Enceladus, but also Mimas and Titan, and the potential ocean moons Triton and Pluto, and also Ceres, which is an asteroid, but that probably formed outside the CO2 snow line, which is the case here. This is the carbonate rich. That's relevant for bodies that formed in the outer solar system beyond the CO2 snow line. And this is inside the CO2 snow line. The CO2 snow line, the water snow line currently is between Mars and Jupiter. The CO2 snow line currently is between Jupiter and Saturn. So that's why, that's another supporting evidence by the samples brought back from Bennu and Ryugu. It was the first asteroid sample return missions, Hayabusa 2 at Ryugu, and then OSIRIS-REx from Bennu. They both find, totally unexpectedly, they found phosphate salts that came from liquid alteration. So these were previously dissolved phosphates that found little deposits in the sample brought back from these asteroids, which are both supposedly formed beyond the CO2 snow line. And this gives now a lot of evidence for high phosphate availability in the outer solar system. If you form a liquid somehow there, then it should be almost set. There should be a lot of phosphate dissolved in there. If you now look at Europa, that's the moon of Jupiter, then we would be below, in the lower branch here, low CO2 availability, because we are inside the CO2 snow line. pH would be between 6 and 8. But it's still, if compared with Earth's seawater, I mentioned it's 1 micromole, it's still good. But of course, the phosphate in Earth's seawater is also so low because life uses it up completely, or almost completely. There was a very recent finding with the James Webb Telescope observations. This is now for Europa, and they found surprisingly high concentrations of CO2 ice on the surface. And this was now reflected in a way to these results that the Europa ocean might be more CO2 rich than we previously thought. And so this will be one of the big tasks for the Europa Clipper mission, to really find out how much CO2 and with that how much phosphate is available on the ocean on Europa. And I can get back to Europa Clipper in a minute. Before I do that, then another recent finding from a reanalysis of data of the neutral gas spectrometer of Cassini. The first results I've shown, they now positively identified HCN, and almost certainly with the limitations of the mass spectrometer, H2O2 and alcohols. And specifically the HCN, the hydrogen cyanide, got quite some attention because it's a very good catalyst for organic chemistry. It's used on Earth many potential pathways for nucleobases and amino acids. So HCN is a very important molecule for origin of life on Earth. And so it's also good that we have it on Enceladus. And you can also do synthetic reactions to form amino acids just with HCN and ammonia. And ammonia obviously is also very abundant on Enceladus because it's about 0.5% of the plume gas is ammonia. So that was also another boost for the habitability of the moon. But this is a slide that was published with a paper here to show the diversity of species that are found in the plume gas. They're all small because they are volatile, but you see the oxidation states vary greatly. The O2, by the way, is a tentative detection, but still, you have a lot of oxidation states, and it's hard to imagine that all these molecules come from the same liquid source. So what they propose in this paper is that specifically these HCN and alcohol. HCN is not stable in liquid water, maybe a few hours, but not for long. So that's why it was hard to imagine that this comes from the subsurface ocean like the CO2 does and the methane. But so they invoke that its source is actually radiolytically produced on the icy surface. So some of the plume material escapes into the E-ring, but most of it actually snows down onto the surface. So you have a lot of organics snowing down to the surface in the ice grains, and then they are subject to radiolytic chemistry UV mostly, UV induced, and then you can produce HCN. And you could also produce H2O2 and oxygen, and it has been shown that the ice that's produced on the surface slowly sinks down into the ocean on the time scale of 10 to 20 million years. So with that way, you can bring these highly reactive components like HCN or even O2 into the otherwise reduced ocean, and with that create a lot of reactive chemistry at the ice-ocean interface. So we have two places that are interesting for habitability. One is at the bottom where the hydrothermal sites are, but also at the ice-ocean interface, a lot of interesting chemistry could go on. And I'm not an astrobiologist, but because of that, I now work a lot with astrobiologists. They just embrace Enceladus and want to talk to us. And they now think that this alkaline hydrothermal vents, that were locations that are suspect to the origin of life on Earth. And so people are speculating that Enceladus, I said it before, now is the most promising place in our solar system to look for life. And missions are already planned. So ESA is now defining a mission to look for life on Enceladus. It was not too much in the press yet, but believe me, it's really happening. And after the ministerial conference in November, I think there will be a big announcement that ESA, for the first time, has a life finder mission to the outer solar system. It's not a spacecraft looking like that, okay, but it will have probably solar panels. But it will has multiple mission phases. It will first sample the plume material and then detach a lander onto the surface that will analyze the snow that lies there. And also NASA Decadal Survey has several mission proposed, not yet implemented. ESA is already a bit further here, but it could be, depending what the Trump administration does. If they now release the New Frontiers call, then we can have Enceladus mission implementation starting as soon as in 3 years from now or so. But this is still a bit in the future. What is more imminent is the missions to the ocean moons of Jupiter. Both ESA and NASA have flagship missions on their way with the main goal to investigate the habitability of the ocean moons. They're not like life finder missions to look for life, like the future Enceladus missions. They are one step back because we don't know the Europa Ocean as good as Enceladus Ocean. This should be done by Europa Clipper to see if Europa also has a habitable ocean in a similar way as Enceladus has. And ESA's Jupiter Icy Moon Explorer has a somewhat wider scope. It's not. It goes to all the other ocean moons, also Ganymede and Callisto. Specifically, Ganymede is in focus, and they will both arrive in 6 years from now, and they're on their way. And we have a successor instrument of the Cosmic Dust Analyzer is on board the Europa Clipper mission. Both mass spectrometers, so the gas mass spectrometer is greatly improved compared to the Cassini instrument, and the surface dust analyzer SUDA is a greatly improved time of flight mass spectrometer from the Cassini instruments. And we probably have plumes here on Europa, but they are not. It's not certain if the spacecraft can sample them as Cassini did, but they don't have to, because a lot of material from the surface of the moon is ejected into space. Ice grains are knocked off the surface from micrometeoroids. They're basically free samples from the surface that you can analyze with a spacecraft at close flybys. And similarly, you have the aggressive plasma environment of Jupiter sputters off a lot of material that you can find in the gas phase or in the ion phase that will be analyzed by mass specs. So we are looking forward to that, and we actually prepare for that. So this is showing you some basic instrument parameters. So the Cosmic Dust Analyzer here, the Surface Dust Analyzer is an instrument that is now on Europa Clipper, and this is an instrument that is developed for a future Enceladus mission. they all work with a similar principle. So a dust grain hits a metal plate at speeds in excess of 1 kilometer per second, and with that the kinetic energy is high enough not just to evaporate the dust grain or the ice grain, it doesn't matter what it is made of, but to ionize a substantial part of the dust grain or the ice grain. That's impact ionization. So you only do that in space. I don't think you do impact ionization on Earth, where you have much better controlled ways to ionize with electrons, electron impact usually. But here we take advantage of the high velocities we have and the vacuum we have, because we have a lot of high voltages here in the grid systems. You can expose it to open space and it works fine. And then you have the ions created from the impact, and then they are subject to a strong electric field here from the acceleration grid that basically drags out the cations or anions. You can change the polarity, and then put them into a time of flight mass spectrometry. So the time of flight of the ion species depends on the ion mass, and with that you get your mass spectrum. And it's really hard to simulate this process on Earth. So if you want to calibrate such an instrument, you would need to accelerate ice particles to 4 or 5 kilometers per second, which is a challenge at the moment, which people are working here in this institute. But in the past it was not possible, and we have an analog experiment that basically was developed by Bernd Abel and Ales Sherbert now in Leipzig, and which we basically adapted for our needs to simulate ice particle impacts. So we have a liquid beam with a typical size of 50 micrometer, and we shoot at it with a pulsed infrared laser. That is a high-speed camera image of the process. So you have 20 pulses per second, but each of the pulses only interacts for a few nanoseconds with the water beam. And the laser wavelength is tuned in a way that you put a maximum energy into the water, and with that you basically have a thermal explosion of the water beam, and the ions you create with this process, and this was the surprising finding, and that's how Bernd and I met first in 2007 or 8, creates the similar water ions than we observe in an ice grain hitting a mass spectrometer. And that's why we now plug in a mass spectrometer here, and then we get very similar mass spectra. So this is a mass spectra of this lab setup, of course with a much higher resolution than we have in the space instrument. This is from Saturn, a pure water ice grain. And we can simulate different impact speeds by tuning the laser energy and changing the delay time. And now this was a working horse for us to interpret the spectra from the Cassini spacecraft. So all the findings I showed before, the different salts, the different organic species, that was only possible because we had this nice analog experiment where we can replicate the spectra from space. And we now go further in the future and prepare a database for the coming missions to Europa and Enceladus, because we have to create our own database. There's no database for impact ionization, because no one else does it on Earth. That's the advantage of electron ionization. You can just look at the database and then you have the result. And so we made some recent progress. That's one paper out of many we did. We can differentiate between iron 3 and iron 2 in different salts. So if we encounter iron 2 or iron 3 at Europa, we can infer different processes at the subsurface. This is some decision tree, depending if you sample it in the blue material or if it's knocked off from the surface. You can already learn a lot. That's just one example, and we now start to mix different salts with organics to see how they would modify the mass spectra. And we are now got funding to look for biosignatures to really see if we can find life in these ice grains, if there would be life in the ice grains. And we showed that we can distinguish, this is now fatty acid patterns that you would have from membranes from cells, just to see if You can create it abiotically or biotically, and you have different patterns that would distinguish both amino acid patterns and fatty acid patterns. So these were done artificially, but recently we also tried to work with real microbes, and the motivation for that was that if we really invoke this organic enriched film near the water surface, then you could imagine that if there's life in the Enceladus ocean, then you would collect some of this cell material here. It doesn't matter if it's like dead or living cells, but if you have cell material either from the hydrothermal sites that is carried upwards, or that uses the chemical gradients you have here at the water-ice interface, if there's life in the Enceladus, the chances are high that they are here, and that in that way they could be entrained in this organic enriched ice grains, which we could not really decipher with the poor mass resolution we had with the cosmic dust analyzer. And there was actually a simple experiment where they put organic film here in a beaker on top of the water and just evacuated the system, and then you have aerosols forming just from this slow evaporation. And they found that you would find probably one cell they found one cell per 10 captured particles, and if we go down with the cell densities to really low values, that 1,000 cells per milliliter, then we would end up with even less cells. But since with the next Enceladus mission, the HiFi instrument could easily detect 10,000 grains per flyby, even that would not be a problem. And we used the Springophyxis alescensis. So there, for the first time, we collaborated with the microbiology department at our university. They provided this. This is a small bacterium that likes to live in cold conditions. That's why alescensis. And they are small enough to fit in these ice grains. And then we shot on the droplets. You can shoot on the water beam, but you can also shoot at the droplets. And we choose the concentration of the cell material in a way that you would have one cell per droplet. And we then went down to 1,000th of a cell material per droplet. And we could still see the biosignatures. This is now the one cell per droplet. And you see that we've identified various amino acids and metabolites here. Most of it is water and salt, but below the inorganic matrix, we could clearly identify the biogenic fingerprint. And we were really surprised how easy that was. And also, if we look in the anion channel between mass 200 and 320 or so, we also find a fatty acid pattern that is from the membranes of this cell material, very nicely, showing that it's a biogenic fatty acids and not abiotic fatty acids. And now if we want to go to Europa, we have to take into account radiation. That's also something that we're doing together with Bernd Abel at the moment. Simulate the effects on radiation, specifically on organic material and also cell material. For now, we have used here this big electron gun in Leipzig with 10 MeV electrons, and the campaigns are ongoing. We have already good results. It's less destructive than you think. I don't have the mass spectra here, but you would think that if you irradiate for a few hours, any organic material will go to pieces. It's not the case. You destruct things, but then you start new chemistry, and you still have larger organic molecules surviving these heavy electron bombardments. So this is good news for detecting organics at Europa's surface. And then of course we take this icy material, it's irradiated, we put salts, organics, or cell material in it, and then we analyze it with the mass spectra. And then finally, we now want to go beyond time of flight. We want to introduce orbit trap mass spectrometry, ultra high resolution mass spectrometry, and that was the first project we had with Jan Zapka here, where we try to connect our liquid beam instrument with a laser desorption, not with the time of flight, but with an Orbitrap. And we had some initial results. So this is now the sketch. To the right, this is now the laser beam, and we could turn the laser either feeding the time of flight system or feeding the Orbitrap system. And then you could also put the liquid beam here or here. In that way we could easily switch between time of flight mode and Orbitrap mode. And it was harder than we thought, but at least we got a few water ice spectra. This is showing the H3O plus peak, and we reached the resolution of 28,700 here. This is totally unachievable with time of flight. So the best we can do with time of flight is maybe 2,000 or 3,000. So this is already 10 times higher, and this is actually bad for an Orbitrap. But it's more than enough. If you could create such a mass resolution in space, you would enter a totally new realm, specifically if you want to detect biosignatures, because then you remove a lot of ambiguity from complex organics. And with that, at the end, got a bit over time, I see. So yeah, we continue to do Cassini analysis, ongoing and future works. We investigate the effects on organic and biosignatures by inorganic compounds and by radiation. We also have, by the way, an ISS exposure experiment to investigate the UV effects. We still investigate a larger variety of biosignatures, and we now try to implement an Orbitrap MS for our analog experiment. And then thank you for your attention.
Okay, thanks for this very nice overview. So I think I always keep on saying, as long as you can entertain the audience, it's okay. So other questions? Oh, yeah.
Yeah, a very interesting talk. So when you investigated this bacterium, how do you know what you want to analyze here as a reference? Is this just something where the biologists say, oh, this is just a probable candidate and this makes sense, or what is the approach here?
Yeah, we learned, until 10 years ago, I had no clue about biology, but like with Enceladus work, you had to embrace this, and then we started to work with microbiologists and astrobiologists, and they tell us that the most abundant cell material is either amino acids, because most of the DNA is made of it, and the second most abundant would be fatty acids that make up the membranes, and that's why we already targeted. And both of these amino acids and fatty acids, we know we can ionize them with impact ionization. So that's good. We tested. We have relative low sensitivity for those. And then the bacterium was also chosen by the astrobiologists. They taught us this is something that you could imagine on Enceladus. And we saw amino acids, fatty acids, and a few other metabolitic molecules. Actually, also some small proteins, fragments. But the lowest sensitivity we have indeed for the amino acids and the fatty acids. Mm-hmm. And they are the most abundant in cell material, so this is very good in both ways. Yeah. But if you, of course, we didn't put any other organics in there. We made our life easy. We put it in pure water. But if you now imagine, okay, we can make it more complicated. We put some more organics and more salts into it. Then the spectrum will be a bit harder to disentangle, admittedly. Yeah.
And the question is always, what is a biosignature and what are you actually looking at? So are you looking at really cells? I think all this is biased against that what can actually be in an ice grain. So this is limited to things that happen at the interface, maybe in a biofilm and so on. So there could be tons of organics, but just heavily diluted. So still the question is what is a sign? What would be a sign of biosignature?
Yeah.
Cell material. Okay, that's.
Yeah, I mean so amino acids you find in meteoroids, right, or in some rocks. But if you create amino acids abiotically, you just create the most simple amino acids, glycine and alanine. They are always the most abundant if it's abiotic. And here you don't see glycine or alanine. The simple amino acids are missing. You see only complex amino acids. And here, histidine, that's something, it's really hard to make abiotically, or arginine, and they are more abundant than the simple amino acids that you would form from an abiotic process. And so you need to detect the pattern of amino acids, then you can tell, okay, this is abiotic or not. But it's not, to find just one amino acid is never a biosignature. You always need the context.
So it's the patterns also of lipids
and the same for lipids, yes. I mean for lipids, you have this typical that you have the that you have C18 and C16 with C17 depleted, for example. That would, because you have this odd. Usually you have the even numbered carbon numbers are more abundant. This C15 here is an outlier. That's a speciality of this microbe, actually. But in an abiotically produced fatty acid pattern, you would have some even envelope and not this up and downs here. Yeah. But it's good to have both amino acids and fatty acids, of course. It always helps to have several. Yes.
Thank you for a very lovely, nice presentation. I wanted to ask just a related question, if there was any focus on like nucleic acid as well.
Yeah, let's see, we have. I mean, we have nicotinic acid. I'm a bit colorblind, so I don't know, but they are less abundant. They're less abundant, so we need they mostly fragment into the amino acid constituents. So we have some small peptide fragments and some nucleic acid patterns, but the peaks are lower. So we simulate impact speeds of 4 to 6 km per second, and with that you fragment amino acid constituents. That's where we're most sensitive to. But of course, if you find a peptide with a mass of 56,000, then this is also a biosignature. But we are not sensitive enough to it. Yeah. It's easier for us to fragment it into amino acid constituents, and then we see the pattern of amino acids. That's also then a biosignature.
What about sulfur? It was just shown in
Yeah.
brackets somehow.
Yes, that's true. For the Schnox, there's this tentative detection of H2S. This is the tentative detection. So the interpretation of the mass spectrometer. This peak it's at mass 34, right? And at 34, of course, you have just another sulfur amino acid. So it's a very tricky analysis of different fragmentation products. And they say this is the most likely candidate, but you can produce the same mass spectrometric pattern. The mass resolution is here just integer mass, so you can just differentiate 33 from 34, but not 34.1 from 33.9. And with that, they can only give a tentative detection. And that's why, but anyway, the phosphorus was the more critical one. And so no one really doubts that there's sulfur available on its atoms. For phosphorus, there was a lot of doubt, but here it's not yet 100% confirmed.
Would it make sense to look?
What? H2O2? Oh, H2O2, yeah. Yeah, I mean, they tested different, there's several papers just looking at how probable that is. H2O2, yeah, H2O2 is definitely present because you have water ice and you radiate it. So you create H2O2. And that's the way how you create, or how you can put oxygen into the ocean. If you have H2O2 in the ice, that then sinks through the ice crust into the ocean, then you have oxidizing chemistry. That's possible here at the ice-water interface. Yes.
Very nice. One naive question. How big must be Enceladus before starting emission of the water?
Yeah, I mean, it's not, so the loss of water, you would think if you lose water over millions of years, then the moon gets smaller and smaller. I mean, that's the point, yeah. Yeah, and people looked into that, but the overall emission is not that dramatic. It looks much more on these images than it actually is. So Enceladus loses 300 kilogram per second. And then you can calculate back. And if you go to it's not clear how old Enceladus is, but the maximum is the age of the solar system, so 4.5 billion years. And if you then plug in 300 kilogram per second, then you lose about 35% of the moon mass. If the plume was as active all the time as it is now, which is questionable. But yeah, it's not a lot. You need to let it go for a long time to make a difference in the overall mass of the moon. But who knows? Maybe it had more water in the beginning and now...
Because condition is different. Because if the masses is bigger, your effect of the pushing of the...
Yes. But there's another factor, this tidal heating. The orbital elements of... so the eccentricity of the Enceladus orbit is changing with time. It's not stable. And this is probably the limiting factor. There was a big paper that calculated back the eccentricity of both Enceladus and Mimas in the past. And they conclude now that the eccentricity is going down from Enceladus. And so the energy input is going down. So Enceladus probably was only active for maybe 50, 100 million years. When it went into an eccentricity maximum, that is like tidal flexing maximum, translating into an energy input maximum, but it's now already declining. So maybe in 50 million years the plumes will go away and then life will die if it's there. No idea. But Mimas, one inward that I put now as a new kid on the block, Mimas has not yet a plume, but has a subsurface ocean, and there the eccentricity is going up, and people suspect that in a few million years the ice will fracture and then the Mimas will have the plume. But we won't know for sure. Yeah.
How is the water coming onto these objects? Is it
It's a building material. So water as a molecule, H2O, is one of the most abundant molecules in space in general.
so
I mean,
just condensation from
and you just have to,
origin of
if you have a protoplanetary disk and you are far enough from the protosun, beyond the ice line, that's why the ice line is so important, then you triple the ratio of solid versus volatile materials. So inside the snow line, the solid materials are mostly silicates, like rock-forming materials. But if you are beyond the snow line, then the most abundant solid is water ice. It's more abundant than rock if you are beyond the snow line. And that's why these icy moons usually have more water ice than rock. It's just the building material that's available. And also the organics get more abundant, as I said. Yeah. If you are cold, if you get colder, you have more condensates. This is all volatile where the Earth formed. When the Earth formed, water was volatile, most organics were volatile.
It's really condensation, not from asteroids coming from...
No. You need this for the Earth. For the Earth, people say, okay, to get the water on Earth, you need to bring it in from the outer solar system, from comets and asteroids. But there, it's already there when you form the moons of the planets. Yeah.
I'm surprised by the hydrogen. which is in principle really a lot, and it has to be very efficient to survive. It's reactive and diffuses easily, so the temperatures are not that low, not that high actually. So it's
probably some of the hydrogen is reacted, but once you release it into the ocean, I mean, it swooshes up like the bubbles that they go up with 10 meters per second or something like that, I guess. But it's plausible. People made the calculation with the serpentinization reaction. You can create hydrogen, but you can only do that once. But that's also a problem with once you have done the serpentinization reaction, it's done. So
it's decomposing water.
It's basically you reduce you oxidize iron in the rock, and with that you release hydrogen from the rock.
Okay.
Yeah, but it's water is also involved. Water is actually the hydrogen source, but it's a reaction of water and rock. But the actual thing is the oxidation of iron in the minerals.
Is there really plumes on Europa, like in Enceladus?
I mean, there are a lot of tentative detections, talking about tentative detections. But people look with Hubble.
I thought with the Webb telescope
Yes, and they haven't found it yet. Have not.
I thought they have some indications.
Some indications, but it's not. I mean, maybe you know unpublished results, but I am not aware that there's now a positive detection. It's definitely less, if it's there, it's less stable than the Enceladus plume. Enceladus plume is always on, all the time. It's sometimes more intense, sometimes less, but it's never like a geyser going down and up. It's always there. And for Europa, they probably are episodic, if they are there. I mean, most people think they are there, but not all the time, because they spend a lot of observation time with Hubble and now James Webb, and but JUICE and Europa Clipper will definitely find it if it's there. Already in the approach phase, already half a year before orbit insertion, both spacecraft will look at Europa to see if they see emissions there with their instrumentation. Yeah. And if we don't have plumes by orbit insertion, then I think they're very difficult to sample, at least. But yeah, we can still have hope. But for the one thing about Europa is the ice crust is much thicker. It's 30-40 kilometers thick. And if you have a plume, you probably don't have a conduit to the ocean, but you have a conduit to some brine pockets that are enclosed in the crust. So to sample the pristine ocean, even with the plume, will be hard at Europa. You probably sample a brine pocket. But it's still a good I mean, brine pockets, why not, right? Good, we'd have some bugs there too.
Other questions?
Yeah, we are almost 1 hour 15.
So just a very quick one. NH4 is surely a mistake.
NH4 is a mistake.
Okay.
Yes. It's NH3. It's NH3. It's NH3. It's neutral gas. Yeah.
But otherwise, great talk.
Yeah. I will correct it right away. Yeah, it's a mistake. Yeah, it's NH3, of course. Yeah. Okay.
Okay, if there are no further urgent questions, of course our speaker is here and you can still ask him. Let's thank our speaker again.